Good morning, everyone, to our webinar on showcasing UC Ventures working on COVID mitigation. My name is Victoria Slivkoff, Global Head of Innovation and Entrepreneurship for the University of California System. Today, you're going to hear from 5 ventures. Each will present for 10 minutes, followed by 5 minutes of Q&A with our expert panelists, Doogie Graham and Christian Bramer. I'm going to turn it over to them to introduce themselves. Doogie, do you want to go first?
Sure, thank you, Victoria. I'm Doogie Graham. I'm the Chief Transformation Officer for UC Health. I act very much as the COO for the whole system of all the medical centres and UC Riverside. So I'm going to be focused very much on the operational aspects and really looking forward to this. Thank you.
Great, thank you. And Christian?
Hi everyone, Christian Bramer. I'm the CEO and founder of Benefunder, and we work with private funders on their philanthropic and impact investment needs, specifically related to emerging breakthroughs stemming from research labs.
Great, thank you, Christian. Great, now I'm going to turn it over to our first presenter, Peter Fronte from Altura, UC Irvine. Just as a reminder, if folks on the phone have any questions for the panelists, please use the Q&A function and we'll try to get to those live, or the presenters will answer them in chat. Okay, Peter, the floor is yours.
Thank you. Let me share my screen here. Victoria, is that up okay?
Yes.
Perfect. Well, first of all, thank you. Thank you for having me, Pete Fronte with Altura, and I'll be speaking about expanding access to COVID-19 studies and patient-centered care. And I'd like to start by thanking Victoria and her UC Innovation Entrepreneurship team for putting this event together and for preparing us so well. So thank you. Okay, so we'll be talking about Altura today, a little bit about our work within the UC system and with academic centers and COVID, the opportunity, the solution, and areas and ideas on how to collaborate. To begin with, there's 2 main issues. One is the speed of clinical trials and studies in general, and the other one is patient-centered care and proactive assessments to drive timely care. On the research front, or the clinical trials front, as you can see here, there's a few key points. Any given year, we're about 2/3 of the population needed for studies short of completing clinical trials on time. That means study sites are not enrolling in a timely fashion, and 86% of the trials are delayed. Related to patient engagement, 5% of the population is driving 50% of the cost, the high acuity patients, and this is exacerbated within the COVID era, and which is why telemedicine is coming to the forefront. However, we still need tools and assessments ahead of the care to ascertain who needs care faster and mitigate transmission by keeping the higher acuity patients from visiting pharmacists or even ERs as an example. So that's the second area that we'll touch on. And just to begin with the end in mind a little bit, the pain points are studies take too long. We don't have enough timely health-related data for high acuity patients. A few solutions are to enable healthcare professionals, or what we'll call HCPs, and all people to access studies on time anywhere, and ensuring that the people and the healthcare professionals have the information they need to act in a timely fashion. 3 ways that we might collaborate. One is Altura is able to support any COVID-19 study or project that requires enrollment or data collection. Number 2 is that we do have an observational longitudinal study that we're making available to employers and medical practices, and we're seeking grants and analytics support there. And 3, we're looking for strategic partners as far as scaling our patient engagement as a service platform. So a little bit about Altura, you can read the details, but I'll tell you a little bit about our passion. We've been around for 20 years, but we're really looking at disrupting 2 industries, the healthcare delivery and the life sciences and innovations industry. What we've seen over the last 20 years is the slow uptake as far as new technology, and a lot of that has to do with only 3% of healthcare professionals being involved in clinical studies. So it's bridging the gap, whether it's a new cancer treatment, an online study like the Michael J. Fox study that we support, the Fox Insight study, or the various UC programs and projects we've done over the years where it was about expanding access to studies and programs in a timely fashion. So that's what we've been able to do, as you can see, with a variety of stakeholders, now utilizing technology, and COVID's a great time to disrupt which is why it's, an entrepreneurial effort here to make sure we include everybody in the innovation and healthcare value-based care model. Just a few examples of our work. As I mentioned, we've done quite a bit of work with UCI, UC Davis. We've had business associate agreements with them over the years to help support enrollment. So a variety of academic centers, Yale, Duke, but also life science companies, foundations like the Michael J. Fox Foundation, but also where the rubber meets the road. IPAs and medical groups that don't have that particular expertise, but we can bring that and help them connect with those experts that are doing these studies. So a little bit about how it works, the opportunity. Most people want to hear from their provider or someone they trust in their care continuum. It doesn't really happen that way. Only 7 in 10 people talk to their medical provider about study options. And as you can see here, historically, there's an overinvestment in direct-to-consumer and an underinvestment with those that are providing care along the treatment continuum, talking about studies. And this is the opportunity to bring studies really to the forefront, especially with COVID, as we'll have quite a bit of a challenge enrolling hundreds of thousands of patients here over the next few months. So the type of research we can support and have supported, as you can see here on the far right quadrant, are the phase 2-3 trials that are going on now for the vaccines and the monoclonal antibodies. But there's various other social and observational studies that are happening and need to happen that may not be FDA directed, but need to happen quickly to improve treatment and improve care. As you move to the lower left quadrant, you have healthcare systems and practices that are looking at quality improvement and practice optimization. All this requires data collection and enrollment, and that's again what we're hoping to provide with our platform. So in summary, the challenges are needing hundreds of thousands of people in these studies to understand and stop the virus. Number one, though, timely access to large volume of people meeting the various criteria is difficult, including ethnicity and comorbidities and testing status. Number two, the study teams and HCPs are very busy and don't always have the tools at the point of care. And three, those organizations, the medical practices and employers, aren't aware of these studies or don't have the tools to collect the data to make that proactive impact on preventing mitigation and supporting high acuity patients. So our HCP Studies platform, we'll talk a little bit about the solution now, is something we created a few years ago, given our experience in healthcare delivery, clinical research, IRBs, HIPAA. Something was needed at the point of care that provide quick access, and this is developed natively in iOS and Android, available in 22 countries, and also a desktop version. And it's about providing quick access to COVID studies, local and online, anywhere, anytime, so people can send information, view information, contact sites, or even learn more about what research is. As you can see here, if you tap on COVID, you will, as I start typing in depression, if I'm going to search, 3 studies come to mind or show up in this particular view. One is a text messaging study, which is about improving depression and anxiety and enhancing positive mood. This particular study, as you click on that study, is actually being done at UC Berkeley, or Dr. Aguilera. And very quickly, we can ascertain what the inclusion-exclusion criteria is. And as you can see, it's fairly open, including Spanish. But once I know what's happening, I need to be able to act, especially if I'm a busy provider. So I need to be able to send study information, contact a team, have my patient with their authorization contacted, or if an evaluation is offered, complete the evaluation. You can see here, clicking on the text or email feature allows me to send information, or anybody to send information to anyone, anytime. I can also contact the coordinator for the study, Rosa Hernandez Ramos, at Berkeley during normal hours or via email to find out more about the study. So we need to facilitate and create more of a viral approach to having studies conducted, whether it's a treatment trial or another type of trial like this that might support patients and innovation. The other aspect of what our platform does is really nimble assessments at the point of care or in a planned fashion that can be pushed out to members or patients to find out more about their risk, their current status with COVID, even contact tracing. As you can see here, this was one medical group that pushed out a survey with high-risk COPD patients, and they were able to get on top of a few people that believed they were exposed, and 21 patients in this particular week that didn't have access to their medications. And these are people who should have isolated. So they were able to proactively deliver the medications and do medication adherence or education to make sure that they weren't showing up at the ER. The last part of what we're doing is an online observational study that's IRB approved, de-identified national information that can be shared with researchers. It's a turnkey study that's available to medical groups or employers that they can post this and have their patients or employers or friends join the study. This is an example of Desert Oasis Healthcare that's listing this study on their website as a way for their patients to participate in this particular study. So in a nutshell, it's about creating a COVID-19 ecosystem, and not just COVID-19. We still need to do important cancer trials, mental health studies, neurology studies. So aligning key stakeholders with local studies, national online studies, virtual assessments to improve care and accelerate innovation is important in what we're trying to do with our platform. So in summary, how to collaborate, I mentioned it up front, supporting any projects that you have that involve increasing program or study enrollment or collecting data. This is all HIPAA compliant in an Azure cloud. Number two, if you have grants available, we'd love to expand this in rural communities with the underserved or people in need that don't typically have access to these types of studies. And three, the strategic partnerships. If someone's interested in patient-centered, value-based care, this is what we've been about the past 20 years and what we look forward to supporting during COVID and when we're beyond this pandemic. So I think we're right at the time. And I know there's a Q&A session. I'm going to bring up the platform so we can take a peek during the Q&A and I can navigate real time to show people, you know, what it's about.
Very cool. Yeah. Thank you for the presentation.
You're welcome.
Do you want to do a demo right now, or do you want to just weave that into the Q&A?
You know, I'll weave it in. I'm
Okay.
interested in answering questions and making sure that I'm aligned with what people are thinking out there.
Great. Okay. Great. Christian or Doogie?
Yeah, thanks, Peter. Great presentation, by the way. And I really like the fact that you're solving, you know, a real problem that can, you know, improve care, accelerate treatments. It's so critically important. I was just wondering, you know, how do you, can you walk us through the engagement strategy? I mean, I also like the fact that you're kind of bypassing traditional media in terms of, you know, recruiting patients. But can you talk a little more about the engagement strategy with physicians and pharmacies and so forth? Like, how does that work? How do you reach out to physicians?
Right, and it needs to be flexible. What we've learned over the years working with academic centers, including the UCI for like the Wisdom study as an example, or some of the phase 3 trials we did with COPD, it depends. The healthcare is delivered locally, and with direct to consumer, it's expensive, and sometimes you get the study sites inundated with phone calls and candidates that may not be the right fit. So what we've learned doing this nationally, whether it's an academic center or a practice, is tying with the research center and finding out how care is delivered locally. And it also depends on the study inclusion/exclusion criteria. So it's usually working first with the study team and the PIs and the sub-Is and finding out who's in their network, who treats these patients, who are the partners in the integrated network or colleagues in the community that have an interest, who might be some of the community centers that could use these resources and their patients can use these resources or studies. So we begin we allow, it's a very easy to use the platform and invite people to participate at no cost and post the studies with IRB approved information. And then again, having 24/7 access to the study to send information. So we start with the study team and who they know and who they prefer to have added to the network. And we also do outreach if that's part of what the sponsor or the study site wants. We can reach out to the community and add users depending on the therapeutic area. One example, I'm working with the Michael J. Fox Foundation, is that they have the Fox Insight study. And while they had a few academic centers as partners, they were really not, the community neurologists were really not engaged with the study. And we developed a national network of 1,000 neurologists and support groups and physical therapists that were on board and were able to have these discussions with patients and caregivers with Parkinson's to get them involved with the study.
Thank you.
You're welcome.
Pete, this is great. Thank you for the presentation. And I think one of the things it absolutely does is I think it helps with some of the blind spots that we see across the University of California, where people are kind of unaware of what's going on outside their own lab or outside their own sort of even medical center. So I think that's a fantastic thing. I wonder, just to make the question of like the kind of the next steps, you know, have you thought how you can aggregate some of the, you know, so that people can start to build on top of each other at all, in terms of making, you know, making the whole greater than the sum of the parts? If you stick with COVID-19, you know, as you aggregate some of these studies, is there a way that you can help with the, you know, as people take some information, they can build on top of each other so that it becomes a kind of peer-to-peer and more collaborative in terms of as people are able to dip in and out of it? Does that make sense? Because I feel that this is a fantastic helping sort of shine the spotlight into areas that people might not be aware of what other colleagues are working on. But I'm wondering how we kind of, you know, can this build upon that and start to aggregate things up?
Yeah, that's a great point, and that's the you know, the point is, and you nailed it, Doogie. Number one is, it just are people even aware of what's happening? In doing research, we've worked with medical groups and even academic centers where there's a great study that's very useful, and across the hall or downstairs, their colleagues don't know about it. And we lose opportunities to not only enroll, but support patients and accelerate innovation. So yes, as part of the platform, Doogie, you know, the first step, as you can see here, I'm kind of driving on the fly, is if you look at, you know, I'm going to bring up a UCSF study, and maybe you're not aware of this. This is an azithromycin outpatient study. That's actually Dr. Oldenburg at University of California, San Francisco. But this can be done at home. It can be done nationally. They mail the study material. It's a virtual study. And this is an example of how we, number one, can have that colleagues around the country that are interested in taking care of outpatient studies that are you know, not hospitalized and receive test results within 3 days. This is important because, you know, inclusion exclusion criteria, you miss that timeline and it's gone. So the timing is everything. And the platform also sends study reminders. Like if this is a new study and we can send that initial reminder, but then we send reminders as often as the investigator wants to the network to let them know, again, the key criteria, like if you have someone that's tested within 3 days, let us know. So this is just one example of how I can, you know, send information or contact Jessica Brogdon. But to your point, as far as building upon each other, once the community is established like this, and it's by geolocator. So if it's a study that's locally and it's only a 30-mile radius because of travel, then we're really trying to draw from providers in that area. But it's a national study like this, then everybody in our entire network and anybody that the UCSF team invites can be included. And there can be discussions, Doogie, related to this study and findings amongst the group, including sharing maybe what's working for a similar study and how they've overcome an enrollment or logistical issues related to operations of clinical trials. So I don't know if that answered your question, but that's exactly the point. Is number one, let's make sure you know about the study. Number two, let's make sure people get involved. And three, how can we share and learn from each other nationally or locally?
Pete, that's great. Thank you.
Great, thank you, Pete. Okay.
Right.
Now I'm going to invite the next team, the team from Konekt, represented by Ravit Sharma, Rahul Guhan, and Santrik Sharma. So,
thank you. I'll share the screen. Is that visible?
yes.
Great.
Oh,
okay.
Okay, it's coming. Yes.
Okay. So let me just go back one slide. Give me 1 second. I'll just restart. There it is. Okay. Hello, everyone. I'm here with my colleagues Rahul and Santrit here. We're Team Konekt, and our solution is space-backed. In a nutshell, what our goal is to provide a video surveillance AI solution that can provide insights by analyzing large crowds in various settings to provide aggregate insights and analytics to local governments with the goal of helping reopen from COVID-19. So thank you once again to Victoria and the team at UCOP for organizing this, as it's a great opportunity for us. So let's get started. Um, So let's start with the problem. If you can take a second to put yourself in the shoes of a public health policymaker, let's say our goal is to make strategic policy regarding social distancing and face covering in order to curtail the spread of COVID-19. So you want to decide on public regulations such as which places can reopen, whether face covering is needed, and even the extent to which social distancing should be in effect, whether it's appropriate to make emergency regulation and such as a shelter in place. So looking at these available metrics that can guide your decision, such as new COVID-19 cases, local ICU occupation, imagine the difficulty in making quick yet responsive legislation in the dark. The problem is that these metrics that are used to determine regulation are delayed and they're not location specific. Individuals are tested positive, they're tested positive days and at times even weeks after the infection. So by the time governments can respond to a new spike in the cases, a significant amount of damage has already been done. And furthermore, the rise in cases cannot be connected to a specific time and location. And so that inhibits proactive enforcement. And the current standard of manual enforcement is error prone and not as scalable as it can be. And studies indicate that 60% of states are not enforcing CDC guidelines. So the current metrics are making it difficult to maximize containment. That's the motivation behind our solution, SpaceVac. How can we remove these pain points and adopt a better standard of enforcement so that these governments and businesses can make better regulations? So we developed SpaceVac with a goal to improve the standard of precautionary regulations and give rise to targeted enforcement and more effective policy. And we do that by analyzing footage from video cameras to provide immediate metrics, location-specific insights, and aggregate analysis of data.
All right, we collaborate with cities and counties to implement our technology into existing cameras at certain locations. And through our subscription-based program, we work with them to set up and calibrate video cameras, understand the specifics of the user, and customize the depth and criteria for analysis accordingly. So the video data from these cameras will be processed at the SpaceVac backend servers, creating real-time analytics on face covering, social distancing, and interactions between people over time. SpaceVac analysis comes together beautifully packaged in a user-friendly and flexible interface that users like cities and counties can use to monitor the enforcement of policy in any particular area. The SpaceVac dashboard can be used to confirm the validity of reported policy violations, as well as getting the specific and reliable answers they need to inform their policy decisions and enforcement. All the helpful analytics and visual maps are displayed on the SpaceVac dashboard. Next slide, please. Thank you. Our technology performs real-time functions such as tallying people in the view of a particular camera, identifying the distance between them, tracking individuals, and whether or not individuals are appropriately masked. The dashboard also contains a 3D and overhead map for a live visual perspective in different settings. SpaceVac also facilitates the development of effective, long-term, data-driven, and even location-specific legislation, analyzing accumulated data to provide cross-location, cross-temporal, and aggregate insights. SpaceVac can provide tailored answers to questions such as what locations in the city contribute most to the spread of COVID-19, or even what time of day or day of the week is enforcement most needed. SpaceVac can also benefit businesses by ensuring employee safety through alert systems for regulation breaches, recommendations for venue capacity, and periodic reports on the business's safety. Use of our technology creates a higher standard of safety for these businesses, which in turn could potentially lead to a boost in profit and growth. Next slide, please. Thank you. Here's a picture of our real-time dashboard that includes a view of live footage, quick statistics for each location, and an overhead and 3D interactive visual display. As a public health policymaker using our technology, you can dive into the real-time analytics useful for enforcement rather than relying on outdated metrics. Next slide. At a glance, SpaceVac provides value because it transforms the metrics to measure the spread of COVID-19 and provides the ability to take advantage of big data, enabling insightful and meaningful conclusions. On the topic of metrics for public health indicators, we will make the transition from delayed metrics such as new cases, contact tracing, and COVID hospitalizations to real-time analytics, such as the number of distancing/masking violations and hotspots. Similarly, we will make the transition from current error-prone solutions such as manual enforcement, Bluetooth wristbands, and smartphone apps to data-driven enforcement and 24/7 surveillance. By providing another set of eyes to oversee crowds, SpaceSpect empowers you as a policymaker to harness data accumulated over time by exposing long-term trends and patterns in the visitation of local areas, as well as the following of regulations. The data-driven answers to these questions allow for the development of agile, direct decisions with the certainty that they are substantiated by specific patterns of public behavior in their area. Next slide. Thank you. Compared to other solutions such as wearable wristbands and geolocational contact tracing, our computer vision-based approach provides unique advantages such as adaptability to new individuals who come in the frame of our technology and is free of personally identifiable information such as perceived demographics or even facial image, so it poses minimal privacy and security concerns overall. Camera and server-side encryption further secures the data. Implementation of our technology is a facile process that we've established through working with multiple cameras.
As a B2G SaaS company, we reach out directly to businesses and local governments who are interested in using our product. In the 6 months since we founded Konekt, we've begun discussions with cities and counties throughout the Bay Area and have refined our solution based on user feedback and discussion. Currently, we're starting our beta pilot program with local governments. Next slide, please. By December, we want SpaceSpect to be implemented in 15 different locations. We're seeking our first seed round of funding of $150,000 to achieve this goal. We will use this fund to reach out to more counties, improve our marketing and outreach, arrange demonstration programs, hire more employees, and continue to enhance SpaceSpect from user feedback. We know that our product would be an invaluable asset to cities domestically and globally. Following these CDC guidelines have proven to be the best ways to prevent the spread of the virus and to make sure we are safe as we can be. However, we can't do it alone. We need your help with acquiring connections and getting SpaceSpect implemented. Next slide. We have a diverse team spanning from machine learning developers to business management. Our contact info is at the top, and feel free to connect with any of us on LinkedIn. As a recap, our solution can transform the speed and detail of the measurement of COVID-19 spread, which would enable higher quality enforcement, more effective long-term public health policy that will allow us to keep safe while we try to figure out a vaccine and more permanent solution to this problem. With this in mind, we're excited to continue working towards our goals, and we're now open to any questions. Thank you.
Great, thank you, Konekt team. I got a question in chat. Have you considered how the public will react to this Big Brother approach?
I can take that one actually. And that's a bit of an interesting question, and we've actually run into that a lot. So I can answer, I have a few answers to that. We've actually designed our tool SpaceSpect with privacy in mind, knowing that we are analyzing the actual footage that comes in this from the security camera or web camera, whatever video camera is set up. Number one, the only data we're collecting is biometric data. No biometric data, no personally identifiable information, merely the locations of people as they move across the screen, and potentially if they're turned towards the camera, whether they're wearing their face masks or not. So kind of the data that we're gathering opens up several doors to insights that could never have been discovered before, such as pinpointing which locations in a particular city tend to give rise to the majority of cases. And it does so without putting any personal information, such as facial image, at risk, so to speak. Another aspect of the way we protect security is through encryption, of course. So camera side, server side encryption, so unauthorized people don't get their eyes on the data if they're not supposed to. And so keeping this in mind, our goal is to provide the most value we can in order to these counties, to these businesses, without putting the public at risk or any personal information, such as potentially license plate numbers or facial image out there for security purposes. And I hope that answers that question.
Great, thank you. Any questions from Doogie or Christian?
Sure. Yeah. First of all, just quick feedback. I like the fact that you're integrating with existing video and surveillance infrastructure. I think that's great. And the predictive nature is just so important to be able to inform public policy and even potentially reduce liability for venues. Just a quick question: what other what other use cases do you see for this technology kind of post-COVID?
I can go again, unless Rahul is interested. Okay, I'll take this one actually. So yeah, there's actually quite a few use cases, and especially in the past decade, we've seen a number of new startups and different applications of how we can use these security cameras that are already there, taking advantage of their 24/7 footage, as well as the advances in AI that have come about recently. So there are several examples. I can give one example of vehicle analysis. So that would also be quite important to cities. And for example, you can go on Google Maps and see what roads are busy, and that comes as a consequence of the fact that several people are using the same app. So it opens up the gate to analyzing this large amount of data. So similarly, what if you have the cam set up as they already are over perhaps a road or a busy intersection? You can find answers as detailed as who is making a right turn along this road, what is the throughput from street A to street B, and even what is the number of pedestrians, how many people are waiting in line, and that can be used for a number of applications, one of them being perhaps optimizing how long people have to wait for the red light to turn green. And so this kind of data can feed into the cities and counties as they plan to make better roads, as well as to get a better understanding of perhaps which roads need to be expanded or need revamping in order to make a better type of infrastructure that can support the traffic.
And if I could add on to that, our technology would also help drive the evolution of smart cities, having more data-driven analysis decisions. If a new business wants to be constructed or anything wants to happen and you need more data on the ground, foot traffic and etc. before you make any city hall decisions, because we've talked with people in city hall and they would really benefit from more eyes on the ground and more before they do anything. They would like to see just on like space A, what's happening here, space B. So that's how our technology would help post-COVID.
Great, thank you.
Sticking with the COVID world, yeah, if I may, I think where this could really help is if you think about, there's a latency question here. If you think about your ability to find potential cases of transmission and likely transmission based on lack of masks, in my mind, I'm trying to think of how do you then link to the modeling and predictive modeling that allows me then to know that therefore in 10 days time, Irvine or Davis or LA is going to be hit with a large number of potential COVID cases. That will be really helpful. Being able to take the latency and say, there were these gatherings, therefore, hey guys, it's extremely likely that in 10 days time, when we do the modeling and parse out the likely numbers, bring it all the way down to therefore likely admissions, that will be really, really helpful. Us being able to actually see some of this coming. And so maybe it's more an observation than a question is, I think you have to try and take this and find a way of then integrating into something else that can do some of the predictive modeling that could say to us, hey guys, you really need to be ready in San Diego or in Irvine because we've got all of this data that shows us that actually you guys have got a bit of a tsunami coming towards you because we had all this data that we saw of people who were maskless and therefore likely to transmit and therefore likely to lead to admissions. That will be really helpful for us, really helpful.
Definitely. And if I can quickly just add on to that, that's actually, as part of the aggregate analysis part we're working on, we've developed as part of SpaceVect. So users such as counties and cities and local governments, they can actually get the specific answers exactly as you mentioned, and even customized to their particular needs. So for example, let's say you have 5 different cameras set up throughout a county. One of them happens to be at the park. Now, when it comes time to figuring out, for example, where is the enforcement needed? Where do we need to have, a couple of police cars or, manual enforcers out there so that they can go ahead and say, hey, and go ahead and control the overcrowding in that particular area. So we can actually use a 24-hour continuous level of analysis to find out and pinpoint not only the locations, even the time of day where the enforcement is needed, and even predict that, extrapolate into the future. So you can have a bit more targeted enforcement, such as where to have the proper enforcement, where to have, what type of regulations you should make, and how is the current status of enforcement of those regulations, so that you have a better cycle of making legislation as well as enforcing it.
Great.
I mean, enforcement's less interesting to me, but I get what you're saying. I'm more interested in seeing things coming towards me from a distance. And so, yeah, the counties need to deal with enforcement, but I'm more thinking about when am I going to make sure my staff are fully staffed, or maybe 80% because of what might be coming at them. But I appreciate what you guys have done. Thank you.
Thank you, Team Konekt. Great. Now, there's a question for you in chat. Make sure you respond to that. Let's move on to the next presenter, Dr. Peruz Kavanpour from Marvell Diagnostics, UCLA. Peruz?
I forgot I'm mute. Sorry. Okay. Okay. Are we good?
Yes.
Okay. Excellent. Let me move stuff from my surfaces. Everything's all over the place. Okay. So thank you very much for UC for organizing this. And also, thank you for Christian and Doogie. I already talked to both of you, so I guess you guys know me fairly well. Doogie, I owe you an email. So let's get going. Marvel Diagnostic is basically a diagnostic collection and diagnostic device to be able to detect COVID-19 along with other respiratory diseases. And we like to call it one breath at a time. So that's our motto. Moving forward, so what is the problem? If you guys are here, you're very well aware COVID-19 is a big problem. Respiratory diseases like COVID-19, influenza, measles are highly infectious and deadly diseases. And what they have in common is actually they're all through the breath, through the exhale breath. They spread. One of the actually bigger killers in the world that not many people talk about is the tuberculosis, also infectious through the breath, and it kills 1.5 million people per year worldwide. Detection and contact traces are necessary to mitigate the economy and the human cost of the disease, as everybody knows. As you can see in the picture, and as you are, if you may have been tested, like I did twice already, the nasal swab is a gold standard for sampling. And as you can see how deep it goes in the back of the nose, is extremely uncomfortable. It's relatively invasive, and also the swabs are special and they're short of supply. And it needs actually a trained professional to run the sampling. Saliva, which is another way, and it's getting more and more traction these days, is lower on a viral load. There are some studies that suggest actually is that because in a saliva is a secondary source of accumulation. So it depends on what stage of disease you are. You may or may not have enough viral load. It's actually a sample quality is not very good. It's as people like to call it dirty. It has a lot of other enzyme and other things in it that actually can cause problem with PCRs. The other thing I would add is actually 2 weeks ago at the FDA town hall, FDA announced that they're not really that much interested in saliva for a variety of reasons, mainly because of inconsistencies that they're getting out of saliva. However, the following week, they actually approve a saliva EUA. So just take it with a grain of salt. Correct detection systems. Let's move to the detection. One of the biggest challenges is slow throughput. We have generally around 1 hour turnaround for the fastest systems commercially available. And limit of detection is relatively large. And at this point, there is not really a point of care system available. So we are trying to sort of address all of these altogether. So our goal is to transform respiratory disease sample collection and detection. Our vision is we're going to use a low-cost, non-invasive collection system and a point-of-care detection device to address this issue for COVID. And as the previous speakers and the question came from the panel, what are you going to do next? Well, COVID may be done, but then there are many, many respiratory diseases that need to be addressed. Also, just add on to that is a lot of people would like to use our system to look into exosome and other markers for cancer, especially lung cancer. So I'm not going to go there, just put it out there for if anybody have any interest to reach out on that part. Our mission is to have a collection system and a detection system for pathogens that exist in exhaled breath, providing both viral load and level of patients. So not only we say positive, negative, they're going to actually eventually say the level of the amount of virus exists in the person exhaled breath, that actually a level of contagiousness on the person. Our solution, I'm going to start talking about the collection system, which we call BlowFISH. Obviously, because you blow into it, and BlowFISH look like coronavirus, just for those of you who might be interested where this comes from. So it's a novel approach for the exhaled breath condensate. A lot of people call it a breathalyzer. I just want to say the distinguished part about what we do. We actually collect virus versus other breathalyzers that exist in the news. You hear about them. They look at the volatile component that's a secondary byproduct of potentially COVID-19. And we are going after the virus. So we are way more accurate because we try to detect the virus. Basically what we do, BlowFISH collect virus directly from lung by what you see in the picture, when a person blow into the cold surface in the backside, the condensation start forming and trapping viral particles. Within 30 seconds, we're going to have enough samples that can be used in any detection system. We are agnostic to the testing, as I'm going to get to it. We can use it, for example, on an RT-PCR. We have multiple patents, provisional patent already filed on it, and on the both condensation and the collection system and the whole point of care system, which I'm going to talk briefly about. So what are the advantages of BlowFISH? It's non-invasive. You're not getting poked. Any person, especially if you think about the kids, if you want to think about adults or people with disabilities, they really would feel uncomfortable getting swab in the nose. Also, getting saliva samples can be tricky. This is very non-invasive. It's literally you're blowing into this device. It's provide a substantial advantage, as I said, for the children and people with need. The price point is actually sub $1 to make. So it's very comparable with nasal swabs. It's rapid. Within less than 30 seconds, you have approximately 200 microliter, which is enough sample for RT-PCR. It's agnostic to every detection system. It can give a sample. It's also determine the contagiousness. There are many cases that nasal swab may show existence of virus, but the person may not be contagious. Same for saliva. This is basically telling you if the person is spreading the disease because you go right to the breath. And also it's a good potential for identifying the super spreader, which are main issues. One other thing is it's actually a clean sample, unlike nasal swab and saliva that has mucus and other things involved in it. We are literally have a distilled water from breath and viral particles in it. So the preparation for the PCRs and other systems that are, it could be to sometime a couple of hours prior to even put the sample in PCR. We overcome that.
Dr. Peruz, I want to interrupt you for a second. Can you advance your slides because we're seeing a frozen screen?
Oh.
Yeah.
Oh. Is it frozen? Can you see it now? Is it on advantages?
No. Socorro, can you pull up his slides and drive it?
Oh, I got it.
Oh, you got it?
I'm
okay.
on a pause. I'm on a pause.
Oh, okay.
Let me see why I'm on a pause here. Why it says I'm on a pause. Okay. Is it good now?
Yes.
Okay. So let me just go back. So this is actually a sample is very clean, as I said. It's agnostic to detection system. Therefore, we can use it with different point systems as a PCRs. We are also developing an on-site detection system, which is FISH, which is a fluorescence in situ hybridization, which we're gonna later in a couple of months incorporate this into the BlowFISH, which we're gonna have a BlowFISH point of care system that I can talk briefly about it. It's higher sensitivity than PCR and high throughput. And our estimation is actually we can have up to 200 tests per hour using this. It's gonna take 5 to 6 minutes to do these tests. We can do pool testing because of the samples are very clean. It's easier to incorporate them together to do the pool testing. One advantage that we have, we can eventually use this system as a sentinel system because we sample air, humid air, so we can use that in air conditioning, can be used in airplanes, airport terminals, hospitals, and so on. And this would be very useful moving forward. Validation, we have done many things for validation. As you can see in the pictures on the right side, condensation has been the volume of condensate on a human testing. We already done that. We use nanoparticles. We easily can detect the nanoparticles in a system. We have all kinds of validation that I can discuss. Also, we already looking for manufacturing companies to build the system for us. Already 3 very highly reputed manufacturing companies want to partner with us to build this. They already have done a pre-design of a system for our clinical study that's going to come up. We have a partnership with SafeFit, which is one of the leading producer of RT-PCR point of care. They have a system that actually can run this for 30 minutes. So they actually provide us with the test to do. There are a couple of other PCR companies who wants to actually work with us. We haven't signed an agreement with them, but it's coming up soon in a week or two. Our clinical trial begin at UCLA Health in September. We're going over, finishing up the little nitty-gritty of things. UCLA is very supporting of this, and they see the value of this. We are already in communication with FDA for our pre-EUA. Our plan is to do our 30 plus 30 negative for EUA, which should be done by the end of September, no later than early October. So we can actually go to market just for the collection part as we discuss. Just to give you an idea about the market, which I'm gonna go through later, I guess. The team, myself, I'm a professor at UCLA, Mechanical Aerospace and Bioengineering. Jonathan Rothstein is a professor at the UMass Amherst and in Mechanical Engineering, and Jeffrey Bird is a professor at Northeastern University of Bioengineering. Just to give you a background, how do we know each other? 3 of us were lab mate 20 years ago at MIT. Jonathan and I were PhD student and Jeff was postdoc. So we have enough skill that cover all these things that as we go forward, we have other team members in the business side and so on. I'm not sure if I need to tell you more about market, just put it out there. It's a huge market for COVID, but even without COVID, it's a big market. The nasal swab was reported to be a $1 billion market right now per year. It's actually, the last time I talked to a couple of experts, they say it's actually hit the $1.5 billion. If we take 10% of that market, just get an estimate, our estimation is right after we get our EUA in October, we can start selling even by 10% of these. We can actually make benefit of $10 million per month just by selling BlowFISH for the detection. There's a huge impact on this. There are a lot of other things that we can do with this, especially now FDA is telling us we should be available to do concurrent testing for flu A and B and COVID because the season is coming and we are prepared to do this because we collect the samples. So whatever it is, we can actually test it. As I said, the strategy is we roll out the collection first. The BlowFISH rolls out probably in October. And while we're doing that, we are already successfully detected lentivirus of COVID using FISH. So we continue on that one. We already have a RADx. RADx from NIH actually funded us for the detection. So we have money from there. We already have an NSF funding prior to this. there are investors already. We actually this week we received 2 term sheets already, but we will see what else we're gonna do. Actually, we're gonna do UCLA has a $250,000 internal grant that we were invited to apply for that as well. Let me see what I'm missing. These BlowFISH are single use and we can sell it to the facilities. I'm not sure how I'm doing with time. So
yeah, I think we need to make time for questions. Let's get to
so I'm
questions.
gonna just stop here with my contact information if anybody want to reach out.
Great.
Thank you very much.
Thank you, thank you. Krishna, since you're already working with UC Health, do you want to say something about next steps?
I'm sorry. Can you say that again? I missed the question.
Oh, I was going to say, since it sounds like you have an upcoming clinical trial with UC Health, do you want to say something about that? Mm-hmm.
I'm sorry, was that for Doogie?
Oh, that was for Doogie. Is he there?
Yeah, no, sorry.
You're on mute.
Am I? Can you hear me?
You're good. Yes, you're good.
Okay. No, I think we're looking forward to this. We've been working with Perus trying to figure out we fundamentally come to the same theme, I would say, over the last couple of days of these is, scale and latency. There's, we're trying to get to doing very large numbers as the campuses look to reopen, as we look to bring many students, staff, faculty back on campus, as we look to have a latency, a turnaround time. This is the direction we've been going in the conversation we're having with Perus and his team of how do we get to, BlowFISH or other things, how can we get some of those things filtered? And also, there's not the trivial question of affordability. If you think about currently the cost per testing, we're in the $100 plus for many tests, and there is no world in which this nation can afford to be testing millions and millions and millions of people weekly at $100 and something a test. No way in the world. So we need to be coming down to $1 or so or less, because that's the reality that we face as we look to a continued testing environment, as we move towards a vaccine. So that's many of the questions and the conversation we're having with Perus, and we, what we're seeing, we really appreciate the work he and his team are doing.
Great, thank you, Judy.
I think just quickly, our point of care we're estimating to be between $10 to $12. So that's something you're shooting for, which is much better than $100. But.
Great, thank you. Very interesting conversation. So let's jump over to the next presenter, Dr. David Sierra from Sanguine DX, UC Santa Cruz.
Thank you for having me this morning. Appreciate the opportunity and some very interesting presentations this morning. I'd like to go ahead and tell you a little bit about what we're doing, if I can. Let's see. Oh, there we go. Share. And let me get to, I can't. Well, anyway, I can just scroll down. Basically, Sanguine DX, this is my 4th company I've started based on my technology. My first company was back in 1992, working with Collagen Corporation, developing surgical tissue adhesives. And over the years of work in biomaterials, surgical intervention devices, and drug delivery and hemostasis, I've moved on from that. I spent 2 years at UC Davis working on some technology for trying to detect food pathogens. And this is stemming from some work I had done prior to that. And here we are back again with COVID, and we decided to go ahead and resurrect the pathogen detection. So we're basically a team of about 12 of us. All of us have been directly involved in the product development. Most of us have products already out on the market in one form or another. And if not, these people have also were heavily involved in developing medical products. We have a lot of experience and a lot of horsepower under the hood. My main collaborators are Solomon Senyange, who just recently sold SpiroSure, a solid state breathalyzer test for asthma, and of course Jeff Christian at Phoenix Ventures, who's partnering with us doing the manufacturing. Again, we have other team members with a lot of experience in developing these things. We're also collaborating with Palo Alto VA Hospital, Santa Clara Valley Medical Center, Stanford, Phoenix Ventures. Our headquarters for our laboratory are at Startup Sandbox here in Santa Cruz, and of course we have the usual O'Melveny, Venable, and Hyman Phelps helping us on the legal side. So what are we doing here? like so many other companies, we're looking at developing a rapid test solution, not only for COVID-19, but also for other pathogens. So what we've created over the last 6 years or so is a piezo-based antigen test using a single-use test cartridge. As you see here in the picture, we've shrunk this down to basically a battery-powered pocketable detector. We've successfully tested different salmonella species. We've tested influenza A antigens, Campylobacter, Ebola markers, and of course some of the coronavirus markers. We have a patent issued for the technology just recently, which is wholly owned, and we're in the process of filing some more start-on patents. I think we can just basically skip this. We know what everybody knows: the main technologies for pathogen detection are going to be molecular and antigen. There are issues requiring sample preparation, long turnaround time for results. Many of these are require trained personnel, lab-based, and they can be expensive and rather resource intensive. What we're trying to address, of course, as everybody knows, the long time to get results, up to 2 weeks. In fact, my neighbor's wife just had to spend waiting 4 days for her COVID test. She was coming down with the flu, and fortunately was able to find out four harrowing days later that she was negative for COVID-19. And of course, has been alluded to before, the inconvenient and uncomfortable sampling, either nasopharyngeal, blood, and of course now more saliva testing. Most of the tests are central lab based and not point of care, which I think is really where I think a major pain point is. And of course, that coupled with the long turnaround time for getting results, I think is really hampering better screening. Of course, there's been a lot of lack of availability of testing for a variety of reasons, affecting not only diagnosis, but also somewhat affecting the vaccine development as well. And of course, there have been supply chain issues and manufacturing issues, just trying to get some of the necessary supplies for testing. So what have we been able to do here with our technology? Basically, we do get a very rapid time for results using saliva. We get it down to 1 picogram per mil. Less than 60 seconds, we have 100% sensitivity and selection so far. We are focusing on saliva sampling for the time being. We are looking at breathalyzer as well, since we have such good sensitivity. Again, it's easy use, minimal training. We're looking at developing this as a point of care device. It's battery powered. We have a pretty low cost for the reader and the test cartridges. Looking at our manufacturing COGS, the reader is certainly under $100 to the manufacturer, and the sampling and test cartridge is under $5 is what we're looking at so far based on large scale manufacturing. The steps to test are very simple. You simply insert the connector end of the test cartridge into the reader. You establish a baseline reading and you remove it. Then you insert the sample well end under the patient's tongue. You collect a small amount of saliva, anywhere from 30 to 50 microliters. Remove, you simply insert the connector end back into the reader, and you have results within 60 seconds. We're doing it, at first we're going to be doing a simple qualitative, whereas coronavirus is, if it's positive for coronavirus above a certain level and indicates green, excuse me, if no coronavirus is detected. This is a quantitative technology. We decided to go ahead with qualitative just to simply get it out the door faster and get this in the hands of people. Basically, so we're doing a very accelerated milestone. We have been in extensive talks with the FDA's EUA department, and we expect to have approval and be market ready, both the cartridges and the reader, by March of next year. We're already scaling up pilot manufacturing at Phoenix Ventures, and we expect to be able to make these cartridges in the millions with a very small footprint. So we've already received seed funding to begin formulation development as well as pilot manufacturing development. We plan on starting clinical trials somewhere at the end of September, early October. Again, dealing with all the usual IRB issues everybody else has to deal with. So we're now started our larger round of fundraising. We're looking at for approximately $3 million to go ahead and fund the rest of the development cycle, and we believe that will be sufficient to get us to market and through approval. So with that, if you have any questions, you can certainly contact me through the Startup Sandbox here in Santa Cruz, or you can find me on LinkedIn as well. And that's what I have for you. I'd be happy to entertain questions.
Great, thank you, David. I'm going to turn to Doogie to get us started.
No, David, that's great. Thank you. Great presentation. And again, it maps against exactly our themes in terms of, you know, cost, turnaround, and just, I mean, maybe one question I've got is, like, do you have a feel for the scale of, like, clearly these handheld readers, you can do quite a lot of throughput, but do you envisage just, you know, how many, you know, could you do a whole cohort of a class, you know, in, you know, what are you trying to, you know, where are you trying to get to with these? Because there is a throughput issue, I suppose, with the, you know, a bottleneck with the actual reader itself. And, you know, clearly you could just have more readers, but you need more people to use it and to, you know, look at the sample and look at the reading. So where do you see this actually as the scale going up to, if that makes sense, David?
Right. Well, we're starting with just actual clinical users. For example, we're working with several anesthesiologists, and they would like to have a rapid screen of the patients before they get rolled into the OR. Obviously, in clinics, the waiting rooms, screening patients before they go in, ERs, that sort of thing. So we're trying to stay really focused on that, because we know we can multiplex the signal into these readers. That would be a next generation where you would simply create an array of these detection samplers, basically like a 96 well plate, and you could just do multiple sampling with high throughput that way in a laboratory. But again, we're just having developed medical products and having to put them on the market in the past, we've realized just keep it simple, go for the most acute market need, and that, to us, that seems to get in the hands of clinicians so they can screen it. Think of it just like a digital thermometer. That's kind of our approach.
Thank you.
Christian?
Yeah, thanks. Great presentation, David. I like the form factor and the fact that it's non-invasive, and the speed to results in 60 seconds is pretty amazing. Just one question I've got for you in terms of your go-to-market strategy. I mean, do you see this? You know, do you see yourselves going after the testing at home market? And elaborate on that, please.
Yes, eventually we would. Again, we're starting, we want to start with a professional use first. We're actually talking at length now with Johnson & Johnson and the Gates Foundation. We're in due diligence with both of them. And so we've begun discussing strategies for how we would roll this out. Again, I like to do it modularly, start with the simplest, start with the expert users, and then build it out from there.
Thanks.
Great, thank you, David. Great presentation. Now I want to invite our last presenter of the day, Dr. William Zinnanti from the Zinnanti Institute of Neuroscience, UCLA.
Thank you. I'm grateful to be invited and a part of this. So thank you very much. Here I'm going to share my screen and get started. There we go. Can everybody see that?
Yes.
Okay, so first a little bit about me. I have 3 decades of experience in medical device development, design, holding 9 issued patents. It was when my son was diagnosed with autism in 1998 that I beelined it to school to seek a medical education, and started that adventure at UCLA in the molecular cell and developmental biology department, where I fell in love with research. I was lucky enough to get a physician scientist training program spot. And after getting the PhD under my belt in that program, I took an invitation for a 1-year postdoc at Caltech to continue research efforts, and then completed residency training in pediatrics and neurology at Stanford. I currently have a medical device business in Santa Cruz known as Zinnanti Surgical, and my clinical practice is across town where I specialize in autism, epilepsy, and neurometabolic disease. My advisory board is really my guardian angels, starting with Professor Larry Steinman from Stanford, where I had a bench and a desk in his lab. Dr. Steinman is best known for Tysabri, which is one of the first biologics and a powerful tool against multiple sclerosis. He's well versed in commercializing bench-to-bedside innovations. Tony Danaher is a terrific role model and friend for 30 years. He's president of Guild Investment in Los Angeles, and he's just exceptional at recognizing an opportunity, but also being able to hold on to the successes of that opportunity because he's thorough with his due diligence, always has been. Nick Hamer is my colleague from the UK. He runs the European sales and marketing for a Swiss software company, Quadratech. He has decades of experience in fundraising, structural scaling, and he's just been a tremendous resource in our efforts to get our idea out. We have an accuracy problem. The current available test, the nucleotide confirmatory test, is showing up to 30% false negatives. The false negatives allow viral spread and make it very challenging for tracing and safety. The accuracy of the serology tests appears to be, the numbers are much worse, some at 50% false negatives and false positives. And this may have something to do with the amplification of inaccuracy from the nucleotide test, because if you're mixing populations that are considered one naive and one exposed to the virus, you may actually be eliminating better antigen targets. And of course, we need a good antibody test to make sure that a vaccine is effective. The current protocols, the most widely used tests, are utilizing one-step RT-PCR technology as recommended by the World Health Organization and the CDC. These tests miss up to 30% of the cases, of active cases. There's too many examples of a positive test after 1 or 3 negative tests, and this has led to the idea that patients can be reinfected because they have been in the hospital. The thought is they've cleared the virus, and then they go home and infect their families and test positive. In actuality, the virus may be hanging out a lot longer than thought, and it probably never left. So the process of reverse transcriptase PCR begins, as most of us know, it begins with the reverse transcriptase of the RNA viral genome targets. And it takes place at 40 degrees, and it takes about 10 to 20 minutes. It's slower than PCR. PCR is much faster, cycling every 1 to 2 minutes. And so the PCR amplification, however, is completely dependent on the product of the reverse transcriptase, so it has something to amplify. If you combine both of these reactions in the same test tube, the faster reaction is always going to steal from a slower reaction and never let the slower reaction finish, and that's where you lose efficiency and sensitivity. So we started out by developing a test in my lab, and we couldn't get the one-step kits. And so this is how I kind of tripped over this discovery, if you will, because I was forced to do the reverse transcriptase and the PCR separately. And I went back to the CDC and World Health Organization protocols looking at the limit of detection, and found that my limit of detection was about 6-fold lower than what they were reporting. Looking into this further, I found a publication from 2009, Al-Shanti and colleagues working in gene expression characterized the difference between the 1-step and 2-step kits that actually lose 30% sensitivity, and perhaps that's exactly matching the 30% loss in sensitivity that we're seeing in the tests. So now we have a problem. Now we have a solution to the problem, but that brings up another problem because, you know, this is the way to put accuracy back in the test, but nobody's going to slow down to add more steps to get this accuracy and do this slower. So I conceived the first dual chamber test tube that protects the reverse transcriptase. And these test tubes can come preloaded with the polymerase in the bottom chamber, with the PCR buffer, and then you would just add the isolated RNA or an inactivated dirty sample with the primers and the buffer into the top. And then the reverse transcriptase is allowed to proceed and complete. And then this proprietary barrier between the chambers melts with a simple temperature change. And the two reactions are allowed to combine, and then this will hydrolyze the probes with a full completed reverse transcriptase reaction, and you get a better signal. So I spent the next bit of time drawing and putting together prototypes, which we then went to submit a non-provisional patent and started doing the testing. Here I'm showing you some of the original prototypes with a dye experiment, and blue and yellow dyes are separated in the different chambers, and as the temperature increases, the blue and yellow get to combine and create green. Here is the results of an actual RT-PCR experiment. And in the top row, we have the two positives, the two control positives, flanked by multiple negative controls. And then you see on the bottom is our dual chamber tube that was able to do this all in one step without more handling of the samples and more steps. So the technology works. It's simple. It's supported by published data, and it puts the accuracy and efficiency back into the most widely used test. The idea can be easily molded, and it can fit all existing equipment. So there's no need to buy a new piece of equipment specifically for COVID. The device can come preloaded, and you just add the RNA to the top with the primers and go. So we have conceptualized the product. We've matured this into a device. We've made the initial prototypes, and we've proved the concept works. And now we've actually made more mature polished prototypes in higher numbers that we're in the process of validating the higher numbers. And what's left to do is higher scale validation and manufacturing, as well as likely, I mean, we can continue this process on our own, but an industry partner with more experience in a space would be of great value to provide the higher number of parts, the validation partners, regulatory requirements, and pursuing licensing opportunities. Thank you very much for your attention, and I'm glad to answer any questions you may have.
Thank you, William. Christian?
Yeah, thank you for that, William. That was a great presentation, and I like the approach that this can be deployed immediately and quickly, and really kind of addresses the speed and accuracy issues, which are so important. Also seems like it solves a logistics issue as well. I was wondering if you could expand a little bit on something I think you mentioned right at the end, kind of related to the go-to-market strategy for this. Once you've secured funding and gone through the next steps, are you seeing this as something that you would create a company and go to market with? Do you see this as a license play? What are your thoughts around that?
100% licensing. For an example, Thermo Fisher has an entire web page dedicated to the differences between one-step and two-step approaches that spell all of this out. And so, a licensing scenario would be the best, as the infrastructure already exists to implement this.
Mm-hmm. Great, thanks.
Any other questions? Maybe talk a little bit about your ideal partners, folks that you like to get connected to.
Well, it was Thermo Fisher and QIAGEN, but now that's one company.
Yeah.
We're being more and more introduced to companies we've never heard of. I believe that Moderna needs this technology, because this is not just for COVID. This is for gene expression. This is for more accurate gene expression, which is currently being done all with one-step kits, and we're not seeing the whole picture.
Mm-hmm. Great, thank you. Thank you for the great presentation. And that wraps up our showcase today. So I wanted to say special thanks to all the presenters for sharing the great work you're driving forward, and the expert panelists, Doogie and Christian, for your terrific questions and helping us to tease out the details. And also audience members, please reach out to the presenters if you'd like to continue that conversation offline. Thank you for joining today. We appreciate you taking the time to learn about all this great technologies coming out of the UC ecosystem, and have a great day.
Thank you.
Thank you.
Great. Bye.